welcome to one more episode of Drug Discovery with Iron Cure. I'm Sukant Khurana. I'm the founder of Iron Cure. With me is Dr. Tejas Gupte. Tejas is the expert of drug discovery at Iron Cure. So today we are going to discuss how plants can be an excellent starting point for drug discovery, for single molecule drug discovery. We are not discussing supplements. We'll discuss supplements in the next episode. We are going to discuss both the pros and the cons of using plants in this episode. Let's start with plants as a starting point historically. Tejas, would you like to discuss some cases where plants have been used and there are modern medicines derived from those?
I mean, let's go from modern to a few hundred years back, right? So the most modern medicine that's derived from plants I can think of is Metformin, which comes from the French lilac, Gallia species. So the origin is clearly plant here. Then there's, of course, Cardiotonics, which are derived from foxglove, Digitalis, right? And we know that they inhibit the sodium potassium ATPase. And even further back, there's salicylic acid, which is the active component of aspirin, which also comes from plants, Spirea species. So three very commonly used medicines, all single molecules, all derived from plants. And because they're all single molecules and they have gone through the entire gamut of testing, preliminary testing, safety testing, efficacy testing, pharmacology, adsorption, metabolism studies, all of that has been done. And we know that they are reliable and they work. And to some extent, they are still derived from plants. So I think thinking of plants as starting material is not something new, and it's definitely it brings a different flavor of compounds into the drug discovery pipeline.
Wonderful. Yeah, I agree. So the thing is that people tend to have this unnecessary dichotomy that they are working with chemical libraries or they're working with plants. Plants do bring in their unique challenges, which we are going to discuss.
Yes.
But at times it takes millions of compounds to be screened and takes years for that screening to find out something which will work. Working with plants has this advantage that if you look at traditional knowledge, then traditional knowledge is not limited to just one or two systems. So people tend to think of, when we think of plant traditional knowledge, Indians usually think of Ayurveda. The better read, they know that it's not just Ayurveda, they think of Siddha system and Unani. If they are East Asians, they think of traditional Chinese medicine or traditional Korean medicine. People don't realize that every single country has its traditional medicine.
Correct.
Not just even each country. If you're looking at a large country like India or China, there are multiple traditions, right?
Yes.
So, in fact, in a large country like Brazil, sometimes you have two different tribes with the medical system that an outsider like me would know more about the medical system of both the tribes than they know of each other. So there are a lot of commonalities. Some of these traditional knowledge might even predate us as Homo sapiens species. We know that chamomile and few other things were found with Neanderthals.
Right.
So traditional these communities get access to them, get them to share their knowledge, and they'll do when they trust you.
Right. I agree with that. And so what basically this traditional knowledge allows someone who's involved in drug discovery to do is, if you can conduct a survey, if you can get these people to share, to trust you and to share their knowledge with you, you have basically cut out a few years and cut out a few hundred man hours or machine hours of screening, because now you already have with you a plant species which is showing you some kind of pharmacological activity. The challenge that the next challenge following up from this is, how do you identify the active ingredient in whatever this plant is, right? And how do you take it further for testing and for developing into a drug? And I think this is where biochemical techniques and developments in biochemical techniques like chromatography comes in, where you have to take the plant and you have to separate that chromatographically into its different constituent